Good morning, everybody. Thank you for inviting me here. It's great to be here. I'm from MD Anderson, where we have a drug discovery center, which I wanted to advertise to you for a few minutes and tell you a little bit about what we do. I think it's a very unique asset that the state of Texas has invested in. And then I want to talk to you a little bit about our efforts to try to develop drugs for Alzheimer's disease. And I'll start with this slide here, which illustrates the problem. There's not a single, as Beth said, there's not a single drug available to slow down Alzheimer's disease or any other neurodegenerative disease. So after many, many decades of research, we still cannot offer the patient any meaningful therapy to slow their disease. So why is this? Why have we failed to deliver a drug to the patients? And one reason you might want to think about is how much do we know about Alzheimer's disease? This graph, whoops, that's not a laser, that is. This graph shows the number of papers published over time with the word Alzheimer's in their abstract. And what you'll see is the first paper published around 1915, that's by Mr. Alzheimer himself, and there were very few papers, less than 100, up into the 40s and 50s. It wasn't until the mid-1980s when President Reagan announced that he had Alzheimer's disease that the interest in the field exploded. Now there's over 130,000 papers. So we've learned a tremendous amount, but it's all been in the last few decades. Just for context, people were receiving chemotherapy in the 60s and 70s. So, we're behind the eight ball a bit. This explosion in knowledge has not led to any meaningful progress in the clinic, however. Aside from the few drugs that treat the symptoms briefly and in some people, there hasn't been any other drug approvals, and that also has not been for a lack of trying. If you watch CBS or news or read the Wall Street Journal, you know that every month or so there's a new high-profile drug failure in Alzheimer's disease. There's been billions spent and hundreds of clinical trials run, all to no avail. So the program we have at MD Anderson is called the Neurodegeneration Consortium, or NDC, because that's a lot to say otherwise. So at the NDC, what we had is a pretty simple idea. Let's bring all those people writing those papers together with people who know how to make drugs and put them in the same room, in the same organization, and let's try to accelerate the pace from discovery to clinical trial. And the idea is that the people writing these papers are brilliant scientists who have new ideas, new theories of Alzheimer's, but they don't know how to make drugs. That's a different field. So we were recruited, I'm from the pharmaceutical industry, so are my colleagues. We were all recruited out of industry to come to MD Anderson and bring that expertise directly to the bench side. And drug discovery is super expensive, as was mentioned earlier. $3 billion a year is a lot of money. So this drug discovery does not work well on an R01 funding cycle. So we've been very grateful to have the tremendous support of a number of very generous donors throughout the state of Texas, notably the Belfer Family Foundation and now the Oscar Fisher Project. Okay, sorry, this is kind of a boring slide, but I'll try to make it somewhat interesting. This is our pipeline. If you're used to looking at this, these are the different drug discovery projects you have in place and what stage they're at in the process. So to the left, this is when you're just validating your idea initially, then you move it into medicinal chemistry, and then eventually it enters preclinical development, which is where you obtain FDA approval for clinical trials. We work in a number of different areas. We're agnostic to the cause of Alzheimer's. We don't have a horse in that race. All we want to know is, do we have a drug that can safely modulate a pathway in Alzheimer's that we believe is important? We'll let the clinical trials take care of the rest. So I'm not going to have time to talk about all 12 of my projects, as tempting as it is. So I'm just going to talk about the most advanced one as an example of what we're trying to accomplish. So the first program the program we've advanced the furthest is called DLK, or dual leucine zipper kinase. And we got interested in this protein because of its role in axon damage and degeneration. Excuse me. So these are old data from humans showing this is an amyloid plaque in the human brain. And what you see here, this little white body right here is a swollen axon. If you remember the talks from yesterday, all the beautiful anatomy of the brain and how precise it is, you can see this axon here crossing this amyloid plaque, and it becomes broken and swollen. Obviously not good for brain function to have your axons do that. This is an animal model of Alzheimer's, and you can see a brain cell trying to extend its axon through the neighborhood of an amyloid plaque and see how it becomes completely dystrophic and swollen. This is a fundamental feature of all neurodegenerative diseases, where you have axons very sensitive to damage, and they react in ways that aren't beneficial to the brain. Interestingly, this is not accidental injury. Actually, the neurons are equipped to sense damage and respond to it in a biochemical process. And the first step in that process occurs when, sorry about that, when the DLK protein I mentioned a minute ago, it senses the injury and sets off a chain of events. And the important ones I want to point out, it leads to the activation of the famous stress protein JNK, which in turn activates the protein c-Jun to go into the nucleus. And if the injury is significant, it will trigger the death of the neuron. So back to what Bess was saying, we want to understand why the brain atrophies in Alzheimer's. We need to stop that. And so this may be a mechanism by which atrophy occurs. From a drug discovery point of view, this is a kinase. For those of you familiar with that area, the kinases are notoriously difficult drug targets. There's never been a kinase inhibitor ever to progress past phase 2 for a CNS indication. So that was a little bit scary at first, but this particular kinase has a lot of druggable properties, which I won't go into at this moment. I just want to summarize a decade's worth of work with one slide. People had shown things like the following to exemplify DLK's role in axon injury. This is the optic nerve crush model. So you just squeeze the optic nerve of a mouse, and a few days later, its retinal cells start to die. And you can see the death that happens, so 80% or more loss of the retinal cells. Correspondingly, the DLK downstream pathway marker, PC-Jun, as I mentioned before, is highly induced in the neurons. If you knock down DLK 70%, you preserve almost all of those neurons, and there's absolutely no induction of this PC-Jun pathway. And you can see graphically those data here. Our question was, is this same pathway that people have been studying experimentally, is it important in a human disease? Is this possibly being corrupted by Alzheimer's disease to drive the death of the neurons? So Virginie Bougier-Prevost in our group, a senior scientist in our group, took on that question, and she developed a method for detecting DLK in brains. And what you're seeing here are the axons that connect your left hemisphere to your right hemisphere. So it's easy to see them. They're all arrayed in an order. And this is from a mouse that overexpresses the human protein tau. And these mice develop tangles, and eventually the neurons die. And normally, I see why everybody else was doing this all before, it's the wrong button. So normally in a normal mouse, this is a normal mouse, there's a little bit of DLK in these axons, but over age it doesn't really change. But in these mice that develop the tau-based Alzheimer's-like symptoms, DLK is hypersensitive to the presence of this pathological tau. Before there's any tangles, before there's any neuronal loss, before there's any inflammation, before there's any cognitive problems, you see DLK accumulating in these axons that later degenerate. So it's a very early sensor of neuronal damage. Well, this is the tangle-based model. What about the plaques? These are amyloid plaques in a different mouse model. And what we did when we looked for DLK is saw DLK accumulating right around the plaques. And it's not in the amyloid itself. It's actually in the axons that are traversing the plaques, as we showed later. This happens in humans, too. We see plaques in humans accumulating DLK in the same way. So this death switch that's been studied in fruit flies and worms and in other systems before actually is activated in the Alzheimer's disease brain. So unfortunately, I'm going to have to condense about 2 years' worth of blood, sweat, and tears into one slide for the interest of time. Our medicinal chemistry team developed very specific potent DLK inhibitors. They have good properties, pharmacokinetics. They enter the bloodstream after oral dosing, pass freely into the brain, selectively block DLK, and clinical trials will start in a few months. We've outlicensed this patent estate to a startup company called Magnolia Neurosciences, who's going to take it from here. So as we also understand, particularly from Cliff Jack's talk about the duration of Alzheimer's and the definition of it, clinical trials in Alzheimer's are a nightmare. So maybe some of the drugs that have been developed would have worked if we just understood how to run the clinical trials better. So what are we going to do about that? Are we going to just do what everyone else has done and beat our head against the brick wall and hope for the best? We decided to try to think of a different strategy to prove that our drugs are on the right track. Being at MD Anderson gave us the opportunity to consider the possibility that we could protect cancer patients from the neurological damage caused by chemotherapy. And what you see in chemotherapy is these are nerve fibers in your fingertips, and these are the nerve fibers of someone who's received chemotherapy. The axons are damaged, they're gone. This causes pain. Likewise, in the brain, you may have heard of chemofog or chemobrain. Chemotherapy damages the axons in your brain as well. So in collaboration with Kobe Heinen, an expert in this area at MD Anderson, we tested our Alzheimer's drug in a chemotherapy model. And what Kobe was able to show was that if you give a mice chemotherapy, it develops pain hypersensitivity. But if you co-dose our Alzheimer's drug, you prevent the pain hypersensitivity. And this corresponds to a preservation of the nerve fibers in the fingertips, well, the paw tips of the mouse. As you can see, normally the chemotherapy destroys the mouse's nerve fibers, just like in human, but our drug prevented that. And I won't go through this in detail, but also it preserved cognition in the mouse. So we gave 3 different memory tests to the mouse. Normally there's a deficit when you give chemotherapy, just like in humans, and that co-administration of our drug completely normalized the mouse's cognitive function. And we measured this using fMRI as well to make sure it wasn't just a behavioral artifact. And we've also done molecular studies that correlate with this. We think, just one last word on the mechanism, we think that the fundamental effect of the drug is to preserve and restore mitochondrial function. That's how it's working to prevent neurons from degrading. So just to summarize, the NDC is a nonprofit biotechnology company, essentially, within the walls of MD Anderson. We serve, our goal is to get academic research more rapidly translated into drug development and into clinical trials so we can speed up the pace of accelerate the pace of discovery for Alzheimer's. Our lead program is blocking a key step in the degeneration of the neurons, and the clinical trials will start soon. And we are taking advantage of our co-location within MD Anderson to be able to accelerate the pace of discovery on the clinical side as well. And with that, I'll stop and pass it over.